How many of you have ever come into contact with someone who's been paralyzed by a neurological disease like this one here? Okay, so many of us have, and you probably wished you could do something to help those people out, right? Well, I've been in a similar situation. I've also seen the devastating effects of a neurological disease called ALS in a family member of mine. I'm sure majority of us have become more aware of this disease now from the recent ALS Ice Bucket Challenge that went viral in the media. Now, whenever I come into contact with people affected by ALS, I always wonder and ask myself, well, what could I do to help these people out? Fortunately, I found my answer in medical research focused at determining the mechanisms that lie behind ALS, because we still don't clearly understand exactly what causes this disease. The approach we're taking on this research is very novel. We think that reactivation of viral sequences found inside our own DNA might be responsible for contributing towards neuronal injury in ALS. Now, these viruses are called human endogenous retroviruses, or ERVs. We think that reactivation of these viruses in neurons may promote neuronal injury. The youngest ERV, ERV-K, is in fact reactivated in neurons of ALS patients, and neurons are the key cells that are damaged in ALS. Now, think about a typical viral infection. Whenever you get a virus infection, your immune system tries to kill the infected cells, right? So similarly, in ALS, the immune system may kill the infected neurons in which ERV-K has been reactivated, producing symptoms such as muscle paralysis because neurons that relay signals for muscle movement have now died. Unfortunately, mechanisms behind ERV-K reactivation still remain poorly understood. So determining these mechanisms is the goal of my master's thesis. We've found that a human protein called interferon gamma, shown here, can activate ERV-K expression in neurons that we grow in our lab. And then what could happen is that you could get an anti-ERV-K immune response, like a typical antiviral response, which may promote neuronal death, causing muscle paralysis. So that's essentially the gist of my project, and this project has important implications for ALS therapeutics because we may be able to use antivirals or drugs that modulate the immune system to control potentially pathogenic ERV-K expression in this disease. And this will give hope to thousands of Canadians currently living with ALS but without any cure. Thank you.